Biology 1107, presentation number 10, cell cycle and mitosis. So what is the cell cycle? The cell cycle is technically the life of a cell, but it's really important that we don't think of it as a birth to death kind of thing, because the cell cycle really begins with an event of cell division and ends with an event of cell division. So what happens in between, from the moment the cell is created to the moment the cell divides again, that constitutes the cell cycle. And in the life of a cell, once it's created, it's going to be formed, it's going to grow, it's going to mature to carry out its intended function, and at some point, it may actually divide again. We will understand there are some cases where cells don't ever divide again, and we'll have a special case for those. So in the next series of slides, we're going to talk about the cell cycle, and then we'll turn our attention to the actual process of cell division. This is a typical illustration of the cell cycle. If you were to Google cell cycle and click on images, you'd come up with 101 versions of this. Some labeled, some not. Some with explanations, others not. So if you do not like this particular illustration, by all means, please Google cell cycle and come up with something that you are going to like. Some of them have plenty of additional information on the diagram. But the bottom line is a cell cycle begins and ends with an event of cell division. And that event of cell division is identified by the yellow wedge at the lower left of the cycle. What follows after that event of cell division is a number of different phases of the cell cycle, one called G1, one called S, one called G2, and we'll wrap up with M phase of the cell cycle, which is mitosis and the creation of two brand new cells. Each of those phases are going to have their own distinguishing features. So I want you to get acclimated to this particular illustration. We're going to have a different illustration that will show us the same exact phases and same exact organization. But like I said, you find that visual version that's going to work best for you. Before we even turn our attention to the cell cycle, however, there's one very important thing I need to mention, and that's the term interphase. The cell cycle is the life of a cell. It begins with an event of cell division and ends with an event of cell division. Now, the far majority of the cell cycle is evidenced by a cell that has absolutely no distinguishing features beyond a normal appearing cell with a normal appearing nucleus. There is absolutely no evidence of activity. So interphase is what I'm looking at. My illustration to the right shows me a cell with a normal looking nucleus, and it's demonstrating no evidence of activity. So you might want to scribble that down. Interphase, cell, normal appearance, normal nucleus, no evidence of activity. Now, beneath that, you will see the designations G1, S, and G2. In other words, the first three phases of the cell cycle, G1, S, and G2, are all evidenced by an interphase appearance. I'm looking at that cell, and if I was looking at that cell under a microscope, and it had the normal nucleus and no evidence of activity going on, I could not tell you whether that cell was in G1, S, or G2. It is not visible to me. There's no visible evidence of what those specific stages of the cell cycle are. All I know is that the far majority of the cell cycle is represented by a cell with a normal or interphase appearance. So I go through G1, S, and G2, and then I come to my fourth phase of the cell cycle, which is M, which stands for mitotic or mitosis, which means cell division. And all of a sudden, the appearance of the cell changes. I now have a cell that does not have a normal appearance. There is no nucleus visible, and I do see condensed chromosomes. That's what I'm looking at. I'm looking at those little red lines on that, and those are condensed chromosomes. That is screaming to me that this particular cell is going to undergo division. As a matter of fact, it's screaming at me that this cell is demonstrating visible evidence of activity. That's in contrast to the interphase appearance of the cell. The mitotic appearance of the cell is one that is not normal in appearance. There is no nucleus, and there is visible evidence of activity. Obviously, only a very, very small proportion of the cell cycle might be demonstrated by a mitotic appearance because cell division is few and far between. The cell is going to be spending most of its time with an interphase appearance, and the only time that appearance changes is when the cell is actually undergoing the division process. Now, there's one other thing on this slide I have not mentioned. You will see tucked right next to phase G1, you'll see in parentheses G sub 0. The reason I put that in there is to make sure that you understand that there are some cases where a cell will not divide. If a cell does not divide, it is said to be in G sub 0. We'll talk more about this in a minute. Just understand that G sub 0 is interchangeable with G1. It's just that G sub 0 is a special designation for those cells that will not divide. And as a consequence, those cells will spend their entire lifetime, that is its true lifetime, from creation to death as a cell in interphase. Well, you'll notice that our version of the cell cycle is different. This is my own constructed version that's going to help us to identify the different phases of the cell cycle, G1, S, G2, and M, and what happens during each one. You will also notice that I've bracketed G1, S, and G2, and I've labeled them with the word interphase. If you wanted to, you could go back to that very first diagram I had of the cell cycle, the one with the different colors. You will notice that there's a big fat arrow that runs around 75 to 80% of the cell cycle, and there is the word interphase in that. The arrow will encompass G1, it'll encompass S, it'll encompass G2. In other words, that significant portion of the cell cycle is evidenced by a cell in interphase. Hence, in this cycle here, G1, S, G2, cells demonstrating interphase appearance. And remember, that's a normal appearing cell, normal nucleus, no evidence of activity. And that changes, though, when we get to M phase, and that's going to be followed by a special situation called cytokinesis, which is literally splitting of the cytoplasm. And obviously at that point, once we get to noon or 12 o'clock on our cycle here, I now have two brand new cells as a result. The first phase of the cell cycle is G1, also known as first gap phase, or sometimes called first growth phase. It is the phase that is characterized by the cell growing, formation of cell structures, and differentiation of the cell until you actually carry out the mature function of the cell. In other words, this is a cell having been created out of an event of cell division, growing, maturing, and carrying out its intended function. Every single cell goes through G1. As a matter of fact, G1 sometimes becomes the permanent state of the cell. I mentioned not too long ago, there are some cells that do not ever divide. Those cells are said to be arrested in a state of G1, also known as G sub 0. G sub 0 is a permanent state of G1. There are cells, for example, certain liver cells, brain cells, and other cells in the body that will never, ever divide once they've been created. Those cells are arrested in a state of G1 called G sub 0. So you notice that the first wedge of the pie there has been set out, and that's indicating that's G1, what we've just discussed. And this is actually a part of interphase. In other words, the cell demonstrates an interphase appearance while it's growing, while it's maturing, and certainly while it's carrying out its intended function. If a cell is destined to divide, then it will enter into the next phase of the cell cycle called S phase, also known as the synthesis phase of the cell cycle. It is where DNA is going to be replicated. In order to accomplish an event of cell division, I need two sets of genetic material. I need two sets of chromosomes. The chromosomes I've got need to be duplicated. So one set can move into one new daughter cell and the other set can move into another new daughter cell. So S phase of the cell cycle is clearly devoted to the process of cell division. It is a phase of the cell cycle that is in anticipation of an event of cell division, and it's not the only one. So once again, we're going to indicate S phase as part of the pie, and once again, we're going to indicate that S phase is also going to be evidenced by an interphase appearance of the cell. Cell with a normal appearance, normal nucleus, no visible evidence of activity. That cell could be doing DNA replication. It could be duplicating its chromosomes, but I cannot tell that just by looking at it. Even though we have been through a process of replicating DNA in anticipation of cell division, that's not the only thing we need to do in order to accomplish cell division. Yes, it's great to have two sets of chromosomes because I need to separate those sets of chromosomes, and it gives me two sets of genes for my two new products. However, in addition to that, I need all kinds of other proteins and intracellular structures that are going to help me to actually carry out the process of mitosis, carry out the process of cytokinesis, carry out the process of creating two new cells where I had one parent cell previously. That's what G2 is all about. G2, sometimes called second gap phase, sometimes called second growth phase, features synthesis or the construction of what I would call mitotic hardware, the actual tools that are used for separating the chromosomes, the actual tools used for separating the cytoplasm. I like thinking of it as like if I'm going to build a birdhouse, yes, I need the wood, but I need a hammer, I need the saw, the glue, the nails, the paint, all the other things I need to actually put that birdhouse together. So at this point, we've been through G1, S, and G2. We understand that G1, S, and G2 all correspond to an interphase appearance of the cell. And of those three phases, S and G2 are exclusively devoted to a process of cell division. Our next phase actually covers the cell division process. All right, we've been through our three interphase phases of the cell cycle. We've been through G1, S, and G2. The cell has grown, matured, come to the point where it can carry out its intended function. I've duplicated my chromosomes. I've made all of the necessary proteins and components necessary to separate those chromosomes. I can now actually perform the event, and that event is called mitosis. We are now at M phase of the cell cycle, and M phase of the cell cycle is evidenced in a cell that does not have a normal appearance. There is no nucleus present. There are condensed chromosomes present, and the cell is demonstrating visible evidence of activity. That's a mitotic appearance. So M phase of the cell cycle corresponds to mitosis, which is technically the separation of chromosomes. You'll remember we can actually use the word mitosis to denote the entire process of cell division in eukaryotes, but now we are using it in a more restrictive sense. We've got duplicated chromosomes. Each chromosome consists of 2 chromatids. Those chromatids are joined together by a centromere. Those chromatids represent equivalent molecules of DNA, and we are going to separate those. So it's going to take a number of different steps. As a matter of fact, the process of mitosis involves 4 steps as we are going to identify them. Some textbooks and some websites will document 5 steps. And then on the heels of those steps, we're going to separate the cytoplasm in a process called cytokinesis. Once we have carried out the process of mitosis, separating those chromosomes, we need to separate the cell cytoplasmically into 2 distinct units, 2 distinct daughter cells. And that process is called cytokinesis, the separation of cytoplasm. It is not technically part of mitosis, which is the separation of the chromosomes. So in separating the cytoplasm, we're going to separate the cell structure, we're going to separate the cytosol, we are going to separate the organelles into 2 daughter cells. And there are 2 patterns, depending on whether you're an animal cell or a plant cell. Animal cells lack cell walls. It's one of the reasons why animal cells have their own unique pattern of cytokinesis. We will call this cleavage furrow formation. We will have one slide devoted to this later on in the presentation. If you're a plant cell, you have a cell wall. And that presents a completely different situation. In your case, that plant cell is going to undergo cytokinesis through a process known as cell plate formation, also documented in a slide later on in this presentation. Since we've been talking about the cell cycle, I think what we'll defer looking at the actual stages of mitosis. And right now, I want to talk about how we actually control the cell cycle. Obviously, cells divide when they need to. And it could be a problem if they divide when they don't need to. So within the cell cycle, I would assume, at least it would make sense to me, that there would be controls that would actually determine whether the cell engages or even completes the process of cell division. So we're going to talk about some of those controls. The main control is one that exists between G1 and S phase of the cell cycle. That is commonly referred to as a G1 checkpoint, or sometimes simply known as the restriction point. See, the idea is that within the cell, we can generate cytoplasmic signals. And those cytoplasmic signals can give us the green light as to whether we might want to divide or not. So it's kind of a form of autocrine signaling, that the cell is creating these signals to essentially give us either the green light or the red light on cell division. So if we get to this big, thick red line between G1 and S, it's like a threshold. It's like riding your bike, and you push your bike up this big, big, tall hill, and once you get to the crest of the hill, it's smooth sailing down the other side. But you got to get over that crest of the hill in order to get down to the other side. Same thing's happening here. We need to get to the top of G1 before we get to S. And if the cytoplasmic signals give us the yes, we continue to S and G2 and then M, and we have ourselves 2 daughter cells at that point. If no, we do not proceed and we remain in G1, or sometimes if it's a permanent state of G1, I think we called it G sub 0 not too long ago. Turns out that scientists have resolved a couple of other checkpoints too, and I've got them illustrated in slightly thinner red lines, one between G2 and M, and one right at the end of M phase can actually be 2 points where we can exert a little bit additional control over whether the cell divides or not. But the primary point at which we determine if cell division will occur or not is this thing called the G1 checkpoint. Since we've been talking about the cell cycle, I think what we'll defer looking at the actual stages of mitosis. And right now, I want to talk about how we actually control the cell cycle. Obviously, cells divide when they need to, and it could be a problem if they divide when they don't need to. So within the cell cycle, I would assume, at least it would make sense to me, that there would be controls that would actually determine whether the cell engages or even completes the process of cell division. So we're going to talk about some of those controls. The main control is one that exists between G1 and S phase of the cell cycle. That is commonly referred to as a G1 checkpoint, or sometimes simply known as the restriction point. See, the idea is that within the cell, we can generate cytoplasmic signals, and those cytoplasmic signals can give us the green light as to whether we might want to divide or not. So it's kind of a form of autocrine signaling, that the cell is creating these signals to essentially give us either the green light or the red light on cell division. So if we get to this big, thick red line between G1 and S, it's like a threshold. It's like riding your bike, and you push your bike up this big, big, tall hill, and once you get to the crest of the hill, it's smooth sailing down the other side. But you got to get over that crest of the hill in order to get down to the other side. Same thing's happening here. We need to get to the top of G1 before we get to S. And if the cytoplasmic signals give us the yes, we continue to S and G2 and then M, and we have ourselves two daughter cells at that point. If no, we do not proceed and we remain in G1. Or sometimes, if it's a permanent state of G1, I think we called it G sub 0 not too long ago. Turns out that scientists have resolved a couple of other checkpoints too, and I've got them illustrated in slightly thinner red lines, one between G2 and M, and one right at the end of M phase can actually be 2 points where we can exert a little bit additional control over whether the cell divides or not. But the primary point at which we determine if cell division will occur or not is this thing called the G1 checkpoint. All right, we're about to take a look at this process of mitosis. First, a bird's eye view. Eukaryotic cell division, aka mitosis in its most general sense. It's a part of the cell cycle. It is the separation of chromosomes. It will be accompanied by a process of cytokinesis. I see a parent cell. I see chromosomes. I see the chromosomes lining up. I see the chromosomes separating. As a matter of fact, look more carefully. You'll see each of those chromosomes consists of two parts, chromatids, that are being pulled apart, resulting in two daughter cells that are genetically identical to each other and genetically identical to the parent cell. But before we get to those steps, we need to talk about some details. Some review, some details about chromosomes, and then we'll actually take a look at how the process of cell division might occur in bacteria. And they don't have chromosomes. And then we'll actually take a look at how eukaryotic cells do it. The process of mitosis, a series of 4 steps, followed by cytokinesis, leading to the formation of 2 daughter cells. So here's the review, and we already know that the majority of the cell cycle is demonstrated by a cell in interphase, normal appearance, normal nucleus, no visible evidence of activity. But once we get to M phase of the cell cycle, that's where everything changes, and the appearance of the cell changes. No nucleus present, condensed chromosomes, visible evidence of activity. And that's what we're going to be looking at in a moment. But one of the things we really need to look at is how do we actually get these duplicated chromosomes? We talked about it in terms of S phase, and I even mentioned the fact that you have 2 identical molecules of DNA for each chromosome. Well, let's see what that actually looks like. Chromosomes, definition, are movable packages of DNA. A chromosome is a single molecule of DNA, contains a complete set of genes found on that molecule. Now, in a human cell, we got 46 of those because you've inherited 23 chromosomes from your dad, 23 chromosomes from your mom. And so those chromosomes exist in your cell. Every single cell contains a complete set of 46 chromosomes. In order for that cell to divide, we have to duplicate those chromosomes. So take a look at our unduplicated chromosome is a chromosome that consists of one set of genes. And if you're a cell in G1, that's all you need. A cell in G1 has a complete set of unduplicated chromosomes. You only need one set of genes, and you can access those genes for all your needs. However, when it comes time to divide, you need two sets of genes. So at that point, we go through S phase of the cell cycle, and S phase of the cell cycle, we said, involves the replication of DNA, and I get two sets of genes. I get two molecules of DNA for every one previously. So if I have an unduplicated chromosome, I'm going to replicate it. And I'm going to replicate it, creating two new molecules connected together by something called a centromere. Now, if you take a look at our illustration, you'll see that those two new molecules of DNA are called chromatids. And the idea is that those two chromatids are genetically identical to each other, important point, and they're genetically identical to the unduplicated chromosome we started off with. So what we've done is we've taken that one set of genes, created two sets, and now we're all ready, because in the process of mitosis, I can take that purple one on the left and move it into a new cell, and I can take the blue one on the right and move it into a new cell. So a duplicated chromosome is all ready for an event of cell division. Be sure that you connect the duplication of chromosomes with S phase of the cell cycle. I know we mentioned it earlier, but now you can actually see why we have these two parts to the chromosome, so we can actually separate them to send one set of genes to one new product and one set of genes to the other. And now in your mind's eye picture, in a human cell, this could be incredibly complicated because, like I said, we got 46 of these, which means you have 92 individual molecules of DNA, and you're going to send one set of 46 into one new cell and one set of 46 into the other new cell. It's going to require a tremendous amount of coordination, which will be evidenced when we actually take a look at the steps of mitosis. So we're about to delve into eukaryotic cell division, what we call mitosis, in a most general sense. Now, mitosis in eukaryotes is sometimes also referred to as asexual cell division. Asexual cell division means it has nothing to do with sperm or eggs or fertilization. Somatic cell division is the term used for animals because animals have body cells that divide this way. And in plants, it's commonly referred to as vegetative cell division. So we got these three synonyms: asexual cell division, somatic cell division, specifically for animals, vegetative cell division, specifically for plants. Doesn't matter which term you use, the process is essentially the same. The cells generated, which are called daughter cells, are genetically identical to each other. They're called clones. They're genetically identical to each other, and they're genetically identical to the parent cell that created them. And so in the typical eukaryote, and it doesn't matter whether it's a plant or an animal or even some single-celled pond organism, its job is to increase the number of cells. Now, at a practical level, if you are a single-celled pond organism, that's going to increase your population size. One cell becomes 2, becomes 4, becomes 8, becomes 16, becomes 32, becomes 64, becomes 128, etc. Or if you're a multicellular organism like a plant or an animal, you're going to accomplish growth of the organism. In any event, when we're talking about mitosis, when we're talking about asexual cell division, somatic cell division, or vegetative cell division, we are talking about organisms that have chromosomes. We're talking about eukaryotes. We cannot use the term mitosis or chromosomes for bacteria, because bacteria don't have chromosomes, and bacteria carry out a process of cell division which is going to be significantly different from that takes place in eukaryotes. Oh, Dr. Canals, I see why you closed the last slide with a comment about bacteria. You're about to talk about prokaryotic cell division. That's a very perceptive observation on your part. Take a look at that bacterial cell. Now, what I remember about prokaryotes is prokaryotes don't have a nucleus. Prokaryotes do not have organelles. Prokaryotes are small and simple. And we also mentioned that prokaryotes have a single molecule of DNA that is a simple circular molecule that is unpackaged, I repeat, unpackaged with proteins. Hence, that simple circular molecule is not a chromosome. A chromosome constitutes chromatin, which is DNA packaged with histones. So bacteria do not have a chromosome. However, I will tell you, sometimes you'll read articles and sometimes you'll read books where someone will mention a bacterial chromosome. Please understand, when they say bacterial chromosome, they're referring to a simple circular molecule of DNA found in a bacterium, but technically it's not a chromosome at all. Sometimes they'll talk about the process of cell division in bacteria as bacterial mitosis. Well, that's not a valid term either, because in order to have mitosis, you have to separate chromosomes. If you don't have chromosomes, you can't have mitosis. So I know that sounds like I'm splitting hairs, but the fact of the matter is when bacteria divide, they do not divide through a process of bacterial mitosis. They divide through a process known as binary fission. Fission means to split, binary means in two. It's a pretty simple process. You're going to carry out an event of DNA replication, and I see that second illustration down, you see my single blue molecule of DNA is now a blue and a red molecule. They are identical copies to one another, and then I will accomplish a split in the cytoplasm, resulting in two new cells. Now, the actual process of DNA replication, I've kind of focused in on it bottom left. It's like what happens is you get what's called a replication bubble forming in the DNA molecule. And that replication bubble leads me to form two strands instead of one. And the two strands will be identical to the one that you started off with. And so if you follow the arrows, it won't take long before that replication bubble stretches all along the length of that circular molecule of DNA, creating two where one was previously. But the bottom line, if I take a look at those two cells at the bottom of this slide, those daughter cells are identical to each other, and they're identical to the parent cell that started them at the top. This is not true mitosis, and it's not a true chromosome, but the results are the same. Mitosis in eukaryotes is a multi-step process. Sometimes folks will include interphase as the first step of mitosis. The reason I have interphase in parentheses here is interphase technically is the absence of mitosis. If you think about it, interphase is the appearance of the cell characterized by G1 or S or G2. The cell is not dividing. So technically interphase is the absence of mitosis. But as soon as you get into M phase, all that changes. So the actual stages of mitosis include something called prophase, something called metaphase, something called anaphase, and something called telophase. Now you can Google each of these and you'll get descriptions and you'll get pictures, and I'm all for that. In each of these cases, there are going to be certain things you are going to need to recognize. You'll be able to, or you should be able to recognize the appearance of a cell in any one of these phases, and you certainly should be able to list the events associated with each of these stages of mitosis. But perhaps the initial thing you really need to do is be sure you know the names of these phases and know them in order. As means of an introduction into the steps, let's just take a gander at interphase. No visible change in the cell, no visible chromosomes, no visible activity. This is the active state of the cell in G1, S, or G2. Even though it's an active state, I can't see any activity at all. Now, I want you to notice one thing. At about 11 o'clock in this diagram of this animal cell, you'll see two little star-like structures. Those are centrioles. You might remember back in the presentation on cells, we said that centrioles are microtubular structures that have a role to play in animal cells, and they will determine the plane of cell division. And I said, once upon a time, those centrioles occur in pairs. So there is a pair of centrioles, and we're going to observe something that happens as this cell goes through mitosis. So let's run down our list of things that we've needed to do before we get to this point. Have we passed the G1 checkpoint? Yes. Have we duplicated our chromosomes? Yes, we did so during S phase of the cell cycle. Have we manufactured our mitotic hardware in order to accomplish this process? Yes, we did that during G2 phase of the cell cycle. So at this point, we're all set and ready to get into M phase of the cell cycle. Alrighty then, we'll start with prophase. Prophase is characterized by three things. In many ways, it's the one most difficult phase to remember in terms of the amount of content. But once you get prophase down, the rest of the steps of mitosis are pretty darn easy. So let's take a look at prophase. We experience the condensation of chromosomes, the nuclear envelope breaks down, and the spindle forms. Condensation of chromosomes means my chromosomes, my molecules of DNA, are going to be packaged into a form that's much more easily moved. It's kind of like akin to packing a suitcase, and you're going to go on a trip. You take all your clothes that are scattered all throughout your room, and you stick them all into one suitcase with a handle, and it's easily grabbed and easily moved. Same thing happens in our cell. We got these long molecules representing genes, but we're going to put them into a form that is a lot more tightly packed and much more easily moved. The nuclear envelope breaks down. You'll see the nuclear envelope is like dotted line there, indicating it's starting to get out of the way, and that's exactly what we need it to do. If that nuclear envelope stays, it's going to prevent our ability to move those chromosomes. So the nuclear envelope breaks down, and the spindle forms. Now, because this is an animal cell, notice I've got those centrioles, and the centrioles have now proceeded to migrate. They're heading their way to the opposite poles of the cell. But you see what's forming between them are these microtubules. And those microtubules will be our agents for moving those chromosomes. So three things you got to remember for prophase. Chromosomes condense, the nuclear envelope breaks down, and the spindle forms. In animal cells, the centrioles separate and begin to migrate to opposite poles of the cell, at which point they will ultimately determine the plane of cell division in that cell. Now, once I get the nuclear envelope out of the way, I can now line my chromosomes up at the middle of the cell. The plane, remember the cell is a three-dimensional object. The plane at the middle of the cell is called the equatorial plate, and all these chromosomes are arranged themselves at that point. We're setting ourselves up to the point of where I'm going to be able to move one set of chromatids to one pole of the cell and the other set of chromatids to the other pole of the cell. In other words, moving one complete set of genes one way and one set of genes the other way. And you'll notice at this point in our animal cell, our centrioles have now established the plane of cell division. The plane of cell division is where all those chromosomes are, and that will be the future separating point between the two daughter cells once we wrap up with cytokinesis. So once we've lined our chromosomes up during metaphase, anaphase, it's time for the sister chromatids to separate. Remember, each one of those duplicated chromosomes consists of two chromatids. We'll call them sister chromatids, and they're genetically identical to each other. Now, once those chromatids separate, as I'm looking at it illustrated here, I've got one complete set of genes traveling to the right-hand pole, and I got one complete set of genes traveling to the left-hand pole of the cell. Our last phase of mitosis is telophase, which is simply prophase in reverse. Chromosomes decondense. I don't need them anymore. Spindle degenerates. I don't need it anymore. And the nuclear envelopes, and there's two of them now, reform. This may coincide with cytokinesis, and sometimes it might not. So, remember I told you once upon a time that cytokinesis can be regarded as independent of the process of mitosis. Because as I have it illustrated here, I see the cytokinesis process, that is the separation of cytoplasm, is occurring during telophase. Well, I have seen cells in anaphase that have demonstrated some degree of cytokinesis. I have seen cytokinesis starting as early as metaphase. So there are some variability in terms of coordinating cytokinesis with the actual process of mitosis. So we've been through all four steps, and the most critical one to remember is prophase, because like I said, those three steps, chromosomes condense, nuclear envelope breaks down, spindle forms, leads to metaphase, chromosomes line up, leads to anaphase, chromatids separate, leads to telophase, prophase in reverse, chromosomes decondense, spindle degenerates, and the nuclear envelopes reform. It's not a tremendous amount to remember, but please take advantage of illustrations in your book, take advantage of illustrations online, and make sure this is reinforced. And it's not just the steps, folks, but make yourself familiar with the pictures, because the pictures show you the actual process. And the more familiar you are with the pictures, the more familiar you are with the process, and the better you'll be able to answer questions concerning these steps on exam. Here's our details on cytokinesis. Animal cells, remember that was something called cleavage furrow formation. And I mentioned the fact that animal cells don't have a cell wall. But what they do is they are going to pinch in half. You will notice that our cell indicates a couple of arrows here where some pinching in is going on. There is a ring of microfilaments there. Now, what do I remember about microfilaments? Microfilaments, Dr. Knaus, are solid rods. They're part of the cytoskeleton. Well, those solid rods actually form a ring around the middle of the cell, and they are going to interact with another type of protein called myosin. Microfilaments are made of actin, and when they interact with myosin, they contract. It's almost like pulling a belt tighter and tighter and tighter. So it's the interaction of actins and myosins in this microfilament ring, like a belt around the center of the cell, that eventually leads to the pinching of that cell in half. Cleavage furrow formation is sometimes also referred to as blastic cleavage. They're kind of synonyms of one another. Now, in terms of a plant, told you that a cell wall is present, and that presents its own situation. Once I've accomplished separation of the chromosomes, and once I've got the reformation of two new nuclei, I need to build a brand new cell wall, a partition smack dab down the middle. So the arrows there indicate the secretion point of a brand new cellulose cell wall between the two new cells, and that new cell wall is called a cell plate. So let's wrap it up. We've accomplished cell division of a eukaryotic cell. We called that mitosis. Mitosis, followed by cytokinesis, yields two daughter cells. Those two daughter cells are sometimes also called derivatives. They are derivatives of a common parent cell. They are cytoplasmically separate, they're genetically identical, and once the process is all done, they are both in interphase exactly like the parent cell was when they started. And so at this point, each of those cells will go through their own individual cell cycle, perhaps to divide again.